Hello and welcome. My name is Dr. Manish Kumar and I am the Vice President for Sales and Projects at BIOMEX. Today I'm excited to have Dr. Jeda Belharzan with me. Dr. Jeda Belharzan is the Senior Scientist and Head of Cell Culture at BIOMEX. I'm going to discuss about circulating tumor cells and its role in cancer cell with Jeda. So welcome to this session of cancer research, Jeda.
Thank you, Manish, for the nice introduction. And I have the pleasure, and it is a pleasure to be here and share insights into CTCs, or cancer circulating tumor cells, and the tumor microenvironment in tumor progression and metastasis.
Amazing, yes. So let's start with the basics. What is circulating tumor cells and what is its significance in cancer research?
So the tumor circulating cells or cancer circulating cells, known as CTCs, are a part of the primary tumor actually. And they have the capability and develop the potential to leave this mother tumor or primary tumor to spread in the blood first, to be there, to feel well, and then to colonize wide organs or spread in different organs which are completely far from the primary tumor. So they have the potential to leave this tumor, like I mentioned. So, and this is really amazing for those cells.
Mm-hmm. So you mean there is a primary tumor cell and the circulating tumor cells are actually part of these primary tumor cells which get detached. So automatically the question comes, how does this detachment happen? And if this detachment happen, I wonder maybe ECM also plays a role, the extracellular matrix, right? So can you enlighten us a bit on this?
Indeed, the extracellular matrix is one of the main points or component of the tumor microenvironment. And I have to tell something. In the past, we just consider the mutated cells as a tumor. And we targeted them and we tried to find a target therapy for them, which has happened and it succeeded in the therapy world. But still not efficient enough because we consider only the tumor as some mutated cells, but it's wrong. So considering the tumor as a complex with all the component and specifically the tumor microenvironment, it's very important. But now, and one of the component, as I mentioned, is the ECM or the extracellular matrix. So imagine those cells have the capability to move, to just be free from their own mother where they have been born, and then just degrade the ECM and search away a mechanism which are very well studied, and live and spread in the blood, use the blood as a vehicle to reach the different organs and build micro metastasis or microtumors there.
Wow, amazing. That's fascinating. So you think the circulating tumor cells and the cancer-associated cells that we study, this is part of the pleural fluid or the ascitic fluid. What are the differences between these two cells?
It's a great question because both of them are in suspension, let's say.
Exactly.
But the cancer circulating cells, they are cells which are at the beginning or the first step for metastasis and progression of the tumor somewhere, another place or another organs. So detecting the CTC in that level or in that point is more efficient to predict metastasis. But for the circulating or for tumor-associated ascites or pleura-associated tumor, they don't reach all the blood. They reach only the cavities around this tumor. It's still not the solid primary tumor or the mother tumor, but it's around. And it's, of course, fluid, it's liquid. And the component of the tumor-associated cells in ascites or pleura, the component is completely different. They have other kind of immune cells, for example, the repressive immune cells, suppressive immune cells, which are very important, how the immune cells are inhibited or are kept far from the tumors to be killed by the T cells. So all this, and they have more macrophages and inflammation macrophages or
So you mean the compositions are different. So the cancer-associated cells have a different composition, and the circulating tumor cells have different composition. And you're talking about detection, like if one is able to detect these circulating tumor cells at an early stage, perhaps one can block metastasis.
Definitely.
Right? And what are the technologies available currently that provides the scientist to, detect these circulating tumor cells at an early stage?
For the diagnostic, it's happened. It's really in the labs. You can detect them. I should add here or mention that we have the CTCs are in a low amount in blood. So to have a high population is really not impossible. It's impossible. And the detection is even one of the challenges. There is some technique which you can centrifuge or filter them by the size and this is one of the technique. The other technique is that when you isolate them from the blood cells completely or the immune cells. But it seems that there is a crosstalk even with the CD40, CD45, for example, immune cells with CTC. So you cannot detect them as well for the preclinical studies, but we can see them, we can isolate them and use them for any diagnostic technique, or the goal is even to study them on the RNA and the DNA level or the genetic level, like using new techniques or high-tech techniques like NGS, next generation sequencing, to identify first biomarkers and for diagnostic, and also genes or markers which can be targeted and then block even these reaching the new metastatic cells. This is the goal.
So one step is to kind of detect, and of course with new technologies like the NGS, one can detect these circulating tumor cells. And the other step...
No, Manish. So the NGS is even to detect and analyze the molecular behavior.
Level,
to detect some gene relevant for their surviving and then target them and then kill all the CTCs before they reach. But for this is the NGS. For the...
So NGS is like just for sequencing, right? Studying
for
the molecule.
sequencing the huge of the whole profiling molecule or genes that could be relevant in the behavior and the survival of CTS and also reaching the new metastatic sites. For the detecting, you can separate them from the blood with Percol, these are gradient density, or you can filter them because they have a several size which you can filter and keep them through a filter, and then you can win them.
So you have specific markers to these circulating tumor cells.
So if
because you mentioned about the crosstalks.
Yes.
So I was wondering if they're like very distinct markers to separate them.
So definitely. So there are epithelial tumor cells. They could have
epithelial
EpCAM,
markers, yeah.
But EpCAM, it means that the cells are epithelial and attached.
Absolutely.
So they're not attaching; they're moving.
They're in
so they lose. Some of them are EpCAM positive, others are EpCAM negative because they did the EMT, they are mesenchymal now, not more epithelial. So but I know from the literature there is also some marker from immune cells like CD45 cells or marker. They could be also positive. So we can use this marker. We can use also the size to filter them by the size, but the amount of the cells, the challenge is how to grow them in ex vivo.
Wow. So after you isolate this small proportion of CTCs, the real challenge is to amplify them or grow them in culture.
Expansion of those cells to study more, and that's why, of course, NGS or RNA-seq or the single cell sequencing is even because you don't need amount of the cells, a lot of cells. You can use a little amount of the RNA or of DNA, and you can analyze it. For diagnostics, is for such techniques, it's enough. For the culturing them, so imagine we could make an expansion of them and then produce some micrometastasis which we could test with the new drugs. So this is a very
interesting area.
Even
so, you think one strategy to kind of cure cancer or cure metastatic cancer in particular would be to kind of target these circulating tumor cells?
Yes, because when the cells like we mentioned before, the cells leave their primary tumor. So they are, so the preparation of the metastatic organ or is not ready to 100% till they reach there. So blocking at this moment, targeting them, it's really something very efficient. If we could study them, have the best therapy, and then target them, then we could avoid the metastasis.
And what are the current challenges in targeting them? Because if you have distinct markers, I feel, with new technologies and CAR T cell therapy, and there's so many other cell therapies, one can target them. So what are the challenges?
So the point is, of course, when you have the CTCs, there is some, mostly it's, I would not say random, but it's depends at what time the patient visit the clinic or go to the, when the patient gets troubles and has to visit the doctor. So this is all depends. Some of them, they reach the doctor and it's really the late stage because
That's true, yes.
the cancer is not painful in the beginning.
Yeah, so the symptom comes much later.
Yes, much later, and it's mostly too late sometimes. That if they, because I wonder, I cannot tell you how long they stay in the blood, but this step is really, I guess, short because this is only like you take the train and go somewhere. You reach this somewhere. Yeah. It's like a transition. This is only a vehicle. So sometimes you have a primary tumor or the patient has a primary tumor. By coincidence, the patient goes to the doctor and he recognizes another problem. And then it's a tumor. But in the case you have already metastasis and it's rich, for example, if we take a breast and it reaches the bones, and reach the pleura because it's not so far, then in this case we have to treat all the organs so there is some efficient therapy, but it depends also on the response of the patient itself, and I would also say the lifestyle of the patient is very important, and I have to focus here because I enjoy or I like this kind of, there is a complementary kind of nutrition. I'm not so familiar with that, but I have not only heard, but I saw patients who have this medical therapy and follow this what we are working on to develop the new therapy, but they completely change their lifestyle and nutrition way. And
so you mean a lifestyle change is very important?
Definitely.
Got it. So I mean, it's quite fascinating. You mentioned about EMT, So yes. I think it would be also important for our audience to know about this epithelial mesenchymal transition. So if you can highlight about EMT, that would be great.
We mentioned in the beginning the ECM. And this is a major component of microenvironment or tumor microenvironment. And this is a robust scaffold. You cannot imagine how strong is it. How strong is it that it protects the mutated cells and it blocks the immune cells to come in. So this is also the no immunity first. And second, to recruit also all the normal cells around to be the slave of this primary tumor. This is all depending on the crosstalk with these ECM. But let me show you exactly as an image that and you can understand it. So this is our ECM. Imagine this is the ECM around. And this is our primary tumor, epithelial tumor, like I said, epithelial, close to each other, of course. Then you have some stroma cells, which are normal, but they worked for that because they work for that, and they call CAF, cancer-associated fibroblasts, but it's not our topic today. And you have some immune cells, let's say, immune cells infiltrated.
But this CAF sounds interesting. Maybe we can make another session on this cancer-associated fibroblasts.
This is a big topic, because they are completely normal and they work for them and not for the body. This is completely another, so they are the slave of the, but imagine this is our ECM with all the macromolecule, laminin, collagens, elastin, and some growth factor. And imagine, let's take these cells. So you have epithelial cells, which detached from all the sides. First, it must be first free from the collective or the cell population. And this is the phenomenon, it's called EMT. And EMT is epithelial, mesenchymal, when they have the transition, they look like this. And they leave. Of course, when they leave, they have to break this wall. These are proteins.
Absolutely.
And this is a big challenge. There is a lot of group scientists who are interested to understand that. If we understand that well, we can block this break.
Yeah, so you stop the...
We stop it here.
Yeah.
And in my opinion, I believe in such procedure, when we understand this is the future, when we understand how to block that, then we avoid, because once these ECM went to the blood, they have a cross reaction with another microenvironment. It's blood cells. This is completely different than that. So you have another challenge to understand first these cross-cell communication. And then that's why it's very important, these EMT. They are like this, but the secret of this, is how to understand the EMT and to block it.
Because it's such an interesting and fascinating from science perspective. So are there ways where, like, you can, because this is much before you can detect the circulating tumor cells in the blood, right? This EMT is going, or is it like the EMT is also an ongoing process? So you have these circulating tumor cells in the blood, and then there's also EMT going on, or it's like totally like initial process?
No, that's right what you said, but it's not called EMT. The EMT is when they leave. They get from epithelial to mesenchymal.
Jera, thank you for explaining EMT. I really liked it, but I have many questions to it. So, I mean, you have the circulating tumor cells in the blood, and this is after EMT has happened, of course, right? So, but is there an EMT also ongoing when you have these circulating tumor cells in the blood? How's it?
So this is, yes, very interesting. Let me go back to a little bit EMT. Like I mentioned, the epithelial cells leave the contact to each other and also to ECM. So degrade the ECM and just leave. And this is the morphology of one epithelial cells got completely different. Of course, this is depending on the molecular profiling. There is some genes very active in epithelial form and other in mesenchymal. And of course, there is, I guess I would not say something wrong, but I guess there is a lot of marker or a lot of therapeutic initiation or therapeutic effect when you target these to block them. But once it's already happened and you have the mesenchymal cells already in there, they are circulating with some, like I mentioned before, there is some marker. They could be epithelial marker have or not. They're EpCAM positive and negative, both of them, because this transition is not like, it's not happening to all the population at once, or well, maybe it's also the epithelial, the EpCAM positive cells are not, they reduce EpCAM.
They lose this marker, basically.
Or they reduce it, that they can be moved and separated and be free and autonomous. But once they decide where they go, they install and become epithelial.
Wow.
So you have not the EMT, you have the MET.
Wow.
So this is a mesenchymal epithelium, and only when you have the MET happening in the sites, metastatic sites, then you have microtumors and micrometastasis.
Wow.
But not all of the circulating tumor cells are capable to do that.
Of doing this.
Some of them are detected from the immune cells and killed. So even boosting the immunity, understanding how the immune cells, when they are in blood, can be, educated to kill them.
Or
it's also an option. Trained to kill them, it's also an option before they really install. When they install in a new microenvironment, a new ECM, a new crosstalks, a new pathways, a new mechanism.
That is so fascinating, Jeda. So because this MET thing is very new for me, the mesenchymal epithelial transition.
Yes, this is.
And is this like, is this well studied that, because you said some circulating tumor cells have the potential to do this MET, whereas some other cells are not capable of doing this.
Yes.
So are there any markers, or are there like any techniques to detect this?
There is some theories against. So techniques I am not familiar exactly. Maybe they are, maybe not. But I know some theories and I'm familiar with some theories. How, now I ask you a question, but just for discussion. what do you think, and what we would think why some tumors go only to brain and not to stomach? Why they go to the bone marrow and not to the brain?
Absolutely. And
This is like they have an address.
they have an address, yes,
So,
certainly.
and this address, there is some vesicles. They are before they do or during the DMT. I cannot tell you exactly. Everything is active and dynamic. So one of the vesicles, this is their address. This is the how to say, the address where they have to go there. So when the installation, when the welcome committee are welcomed in the new organs and prepare everything, the CTCs, these circulating cells, just can attach and find the microenvironment ready or the minimum, and then of course do the MET.
Wow.
And that's why the ones who do not have or failed in getting this address or whatever or preparation because of the immunity, because of some therapy, whatever is it, then they are there. Some of them, they are there, they never fix the organs, but they're still there.
They just circulate, but they're never able to find this address.
Yes, because the preparation didn't happen.
And I think, yes, and absolutely. I think you mentioned one thing which is very important for this localization of this, circulating tumor cells, and that's the tumor microenvironment. So what I understand is like you have the primary tumor, and then you have the detachment of these circulating tumor cells from this primary tumor, and then back to forming microtumors where the TME or the tumor microenvironment is correct. And please, for the sake of audience, what is this tumor microenvironment? And when you say correct tumor microenvironment, what do you mean by that?
So maybe the word correct was not correct, but anyway, I mean with that if, let's take an example that everyone can understand it. So imagine we have a cancer circulating cells from the breast and they move to bone, and you have a bone metastasis. So if you have a bone metastasis, and you have to adapt with all the microenvironment of the bone. And the bone anatomy is completely different than any other organ.
Absolutely.
So, and then when there is a right adaptation or a right preparation first, and adaptation for these mesenchymal cells, circulating cells, or tumor cells who decide to go there and install. So if there is no crosstalk, or if the crosstalk do not or does not happen correctly with pathways and with regulation, so it could be there is no micrometastasis. But if everything is perfect and the preparation of the new microenvironment happened in the correct way, what are the mechanisms is depending on the pathway active in bones or in brain or in liver. This is one of the metastatic organ, so it's really depending on the molecular profiling and the gene active in these microenvironment and different organs. So
fascinating. Jera, amazing. So what I think is, like, I'm just wearing a scientist hat and thinking that, basically there could be 3 strategies to cure cancer. Of course, it sounds childish, but because it's very complex. But basically, there could be like either you target a therapy that targets this EMT, the epithelial mesenchymal transition.
Yes.
Second thing could be also to target this circulating tumor cells, right? So you just block the transition or block the metastasis itself. And the third thing I think is also like one can design therapies where you can block this TME, or maybe I'm wrong, like this tissue microenvironment, or TME. So maybe, for the audience, you can tell us what TME is and whether it is realistic to target TMEs. And apart from that, what could be the best strategy to kind of cure cancer?
So this is the 3 points you mentioned are very important. Exactly. There is a lot of study and a lot of therapy options in these 3 points. The EMT or the MET, of course, definitely. And the first one is, I think it's the research is really ongoing on that, understanding the EMT, because it's irreversible. What I can say is there is no model to study EMT, MET ex vivo. Something is missing in our preclinical study. experimental medicine. Maybe there is some, but it's still really in the beginning. I have not have heard, and that is always a problem. How when we want to target EMT and MET, we have to study more this phenomenon, understand all the molecular level, what happened, and also the environment, like you said. So it's the third point I mentioned. There is some addresses around the sites, metastatic sites. So understanding how the addresses, how these vesicles are moving for the tumor and going to the brain or the bone or the liver, this is also an option. If we understand what are the genes responsible of that, it could be no proliferation of any cells left the primary tumor. But I have to mention something which is really not so advanced, but really current, is again to the mother. If you go to the mother or to the primary tumor, do you now manage that the tumor itself doesn't like if the EMT happened?
Wow, I didn't know this. Yeah.
So, and there is a phenomenon, it's called anoikis. Anoikis is a kind of cell death. So
You mean like apoptosis?
It's like apoptosis, but it's involved the ECM. Apoptosis is involving only the cells, but anoikis is involving the integrins, and integrins are molecules belongs to the
It's a cell adhering molecule.
to the ECM. So, and imagine these primary tumor, like their epithelial cells and daughter cells. It's like a mother and babies around her. When once grow up and do the EMT, the tumor itself induce anoikis and kill them. So
within the tumors, just to understand it better, so within the tumor cells also you have something equivalent to apoptosis, which you call anoxis, right?
Yes.
And this is also a programmed thing like apoptosis, or it's different, or it's like
it's like in the beginning, or the inductor of anoikis is coming from the ECM. So the ECM doesn't like to be broken and left. So it's induced through integrins. The indication or just an inductor, this is intergreen alpha and beta, and these integrins and tubulins, they are activating some genes, and at least you have caspase activation.
Wow.
Yeah, it's like the end product is always caspases, and this is what we do not understand well now.
Fascinating. And from the assay point of view, like, because we have a lab, we do a lot of assays also inside our lab. So are there like specific assays to differentiate between apoptosis and say anoxis? And can we perform these assays in our lab at BIOMEX, say?
So we do not do it now, quite now, but with our homogel. And I think the people include now these matrices, these are commercialized matrices. That's why we have developed the homogel, which is from the human tissue, normal tissues. And of course, we include this component of the tumor microenvironment or for any environment, microenvironment, in our experimental medicine models. So, and I think it's quite now it's used that you co-culture the cells with any ECM scaffold, and of course in the best case you culture them, and then you just give drugs, and or when you have drugs with anoikis drugs, there is some like
you mean drugs that target anoxis?
No, that's
induce exactly, because that will stop, that will block. Yes.
Anoikis inhibitor. So you target anoikis inhibitor, and then you see what happened till you. And of course, this is a model how with the future, using homogel or whatever as a scaffold in 3D, as you have some drugs, a blocker of anoikis inhibitor. So if we could help this primary tumor to induce anoikis, kill all the mesenchymal cells on the way of EMT transition, and then block them, and then we have to deal with one enemy in one location.
Wow, that's fascinating. So, Jenna, you mentioned that there are significant challenges with the current ex vivo model of, cancer. And what do you think are the real challenges in building these ex vivo models?
Yes, I mentioned that, and this is very extremely urgent, and there is a lot of big needs of these scaffold. Because this is a new fashion to understand more tumor biology in 3D. So culturing the tumor cells and all the subpopulation of one tumor in once and recapitulate the tumor ex vivo in the lab. It's a challenging. One of the components we study, you see in our talk now, that the ECM is very, very, very important component in the microenvironment and can be also targeted in the future. And of course, we need this scaffold as a stable, resistant, mimicking the in vivo situation. And what we have on the market are some a lot of plant-derived or animal-derived or subtypes of tumor subtypes derived. And this is not really, you cannot study first the human tissue. You cannot study even the transformation of the first transformation happened on the level of these ECM, and that's why we have extremely, it's urgent to develop a new specific, normal tissue specific, and also tissue specific and species to stay in the human, of course. And that could be very, when we succeed, then I guess we would also have a good opportunity to understand the tumor biology, to understand anoikis, to target it, to induce anoikis, to block the EMT, block MET, and so far and so on.
Amazing. So, I mean, rightly so, because, you know, most of the researchers that I know, they always use gels that are coming from mice, and especially this sarcoma tissue from the mice. And this is already a flawed model because, first of all, it's mice, and second of all, it's from tumor, right? So, you think there is a real need of having a ECM that is derived from normal, healthy human tissues?
Definitely.
One question that I always have, you know, I have friends who went to oncologist, had this cancer treatment. And one puzzling question that I have is about the recurrence of cancer. So as a scientist, why do you think the cancer recurrence happen? Even after treatment, even after cure?
Yes, this is my favorite topic, really. And this is a fashion, the resistance of the cancer. We have two kinds of resistance. We have the resistance where the therapy doesn't work. This is really the best things. We have to look for alternative therapy. And when you see in the clinic, there is not only the oncologist deciding what the patient had. There is a tumor board. There's a couple of clinicians sitting together and take a decision. There is a light line and the recommendation of the therapy. It's complicated. But once I have to mention here that, you know, you have, when you hear the word cancer, you always think and growth and something active and growing, and there are no homeostasis. And of course, we focus on the blocking this proliferation, which is an option, of course. So blocking the proliferation, that the tumor cells just die.
Stop growing, yes.
It stop growing. It's also a really beautiful way and option to cure cancer. But in the same moment, we recognize the tumor is heterogeneous. It's not only a couple of mutated cells, like I mentioned. And there is some cells which they called cancer-initiating cells or cancer stem cells. And they do not grow fast. They sit and look at the tumor and try to, and they are there and have the role, like their name is cancer-initiating cells. They have only the role to support the renaissance of the new cancers. So they are not our therapy or the therapy used in our time are not targeting these cells, and they are dormant cells, and these dormant cells, they're not dying. They are there. They are faced to this stress of therapy, but they resist. They don't develop.
So you mean the cancer initiating cells or these cancer stem cells, so-called cancer stem cells, at the site of the primary tumor are resistant to this therapy. Even chemotherapy or radiation, are they resistant?
Definitely both of them. And I will explain to you why is it like this. So the dormant cells are cells which are I would not say senescent, but it's like a pseudo-senescence. Are you familiar with the word senescence?
Very much. In fact, I worked on senescence.
So the senescence is not reversible.
Yes, it's a permanent cell cycle arrest.
So, and it's one way. But they have the pseudo-senescence because they can reactivate again, and where they can differentiate, and the new tumor is born. And this is what happened. So let me show you one thing. So you have here inside our tumor, you have some tumors. Let's do like this. And these cells are cancer stem cells.
Yeah.
So cancer stem cells.
And these are in dormant stage.
There are dormant cells. When you treat, when you have irradiation, or you have chemotherapy, all these cells can die. Okay? But these cells, they are there and they sit and they have no behavior and you cannot detect them even.
Got it.
But once they are responsible of the resistance, either they are resistant to the therapy because not targeted them, or these cells are dying to, to support the others.
You mean to protect it?
No, to support them. I don't know whether this killing is a normal killing or an autophagy. You know what autophagy?
Of course.
It's a kind of, for the cells. So they make the metabolite free to feed these cells.
Oh, okay.
They are feeded. They have not to be.
So in simple language, they sacrifice themselves to feed the dormant cells.
This is the word. I wonder, me as a scientist, I wonder whether the therapy is inducing a sacrifice for those cells. But this is still enigmatic. I cannot prove it. So once we need that these cells also die, and this is what happened, and this is a success for the patient, of course. But the dormant cells are responsible for these comeback recurrences. Or these cells, or there is another point which we have some genes, like you know when we have a bacteria, there is a lot of bacteria which we call multi-resistance. And imagine, Manish, we have such cells also in cancer, multi-drug resistant cells.
Wow. So Gerard, thank you so much for explaining all this, and I can go on and on like a child, asking you questions, but just to respect our viewers' time, this is time to say bye-bye to all of you and see you in another episode of our scientific discussion at BIOMEX. Stay tuned. Till then, bye-bye.
Thank you very much.
Thank you.
BIOMEX, empowering scientists.